Well, hello everyone. Welcome to the PGC Worldwide Lab. We'll just wait a few more minutes for more people to join. Okay, well, I think the numbers have stabilized. So as I say, welcome to this PGC Worldwide Lab webinar. I was asked to organize this and to focus on methods. And so I didn't go very far. I went to my own group to have two presenters who I think have done some work which I think is relevant to the Psychiatric Genomics Consortium. So first of all, we're going to have Xiaotong Wang and then followed by Zeng. So Xiaotong Wang, we also call him Mark, is going to go first, and his talk is going to be shorter than Zeng's talk, which is coming second. So Xiaotong Wang is he's actually a DPhil student or PhD student here with me in Oxford. He's moved with me from Queensland. While he was doing his medical doctorate degree in Queensland, he did some research with me. But the work he's presenting today is his first work as a PhD student. So Mark, if you'd like to share your slides and start your talk. Thank you.
Right, so can you see my slide?
Yes.
Yep. Right, so hello everyone. So thanks for joining the webinar today, and special credit for those who live in Asian Pacific regions. I know you have to get up really early for this meeting today, so hopefully it will be helpful and be worth your time. All right, so my presentation today will be on polygenic risk predictions, where we explain why and when out-of-sample prediction R-squared can exceed SNP-based heritability. Right, so to test whether a phenotype is associated with genotypes in a certain population, a common thing we do is to collect a discovery cohort from the populations and get their phenotype and genotype data, and then we can do an association test. And with the resulting GWAS summary statistics, we are able to generate polygenic predictors and predict into a cohort that is independent of original discovery cohort, which we named the target cohort. So to measure the performance of polygenic predictions, a commonly used quantity here is the out-of-sample prediction R-squared, which is defined as the proportion of phenotypic variance explained by the polygenic scores. So in theory, the SNP-based heritability in discovery cohort where polygenic predictor was generated is the upper limit of R-squared, because by definition, H squared is defined as the proportion of total phenotypic variance attributable to all common SNPs. And because in generating of polygenic predictors, we just use a subset of all common SNPs. Plus, there is unlikely for us to sample like every single individuals in the population. So that's why H squared should always be higher than R squared. And additionally, sometimes the sample size of a single discovery cohort is not large enough. Another common thing we do is to do a meta-analysis where we combine information from multiple cohorts together. And with the resulting meta-analysis summary statistic, we can also generate a polygenic predictors. And in this case, the SNP-based heritability in the hypothetical meta-analyzed cohort should be the theoretical upper limit of R squared. So that's what we expected. But in reality, we observed some very interesting phenomena. So 10 years ago, PGC estimated the SNP-based heritability in a meta-analysis of 18,000 samples for major depression, and estimates was 21%. So back in 2013, where we expect the SNP heritability in meta-analysis to be with an increasing sample size is that it should be fluctuating around the true SNP-based heritability, but with an increasing sample size, the estimation error should be smaller and smaller. But very interestingly, like 5 years later, when in Wave 2 study, when we re-estimate the SNP-based heritability in the meta-analysis with around 500,000 samples, we realized the SNP-based heritability dropped to around 9%. And now another 5 years later, so in the latest Wave 3 study, we re-estimate the SNP-based heritability and found it further dropped to around 6%. And in parallel to these findings, we also found that in some of the cohort, out-of-sample prediction R-squared can be significantly higher than its theoretically upper limit. So our work is motivated by these observations, and our work is trying to understand why this phenomenon is happening. Right. So first, let's focus on the SNP-based heritability in the meta-analysis. First, we need to define what is a meta-analysis, and let's take a single SNP K as an example. So the true SNP effect of SNP K in certain cohort, like for example, beta 1K, is never observable because, as I mentioned previously, we are unlikely to sample every single individual in the population. So what we are able to observe is the beta hat or observed SNP effect, which can be obtained from GWAS studies. So in meta-analysis, estimated SNP effect for SNP K is simply a weighted sum of observed SNP effect in each cohort included in the meta-analysis. And so by expanding that equation I showed and playing some mathematical tricks, we are able to get an expression of SNP-based heritability in terms of weight applied to each cohort times genetic correlations between all pairs of cohorts times the square root of SNP-based heritability for each cohort. So in real practice, we can estimate the genetic correlations and SNP-based heritability with either individual genotypic level data or summary statistic level data. And the next question is, what is the weight for the meta-analysis? So the weight of the meta-analysis depends on the underlying model assumed. For example, if we assume a fixed effect model where true beta in all populations are same and observed differences are purely due to sampling variation. So if that is the case, then we can plug in the inverse variance weight here. And in a GWAS study, it can be approximated by the numbers of individuals in a single cohort over the total numbers of individuals. And alternatively, we can assume a random effect model where true beta of different cohorts are different. And the beta of meta-analysis in this case should be interpreted as the mean effects of the population of true effects. And in this case, we should add a term here to account for between cohort heterogeneities. So for most meta-analysis for GWAS study to date, we assume a fixed effect model. So the underlying assumption of the fixed effect model is that we are living in an idealized world where each contributing GWAS cohort is a random sample from homogeneous idealized populations. And if that is the case, then the true genetic correlations between all pairs of individuals should equal to 1, and the SNP-based heritability of each individual cohort should all equal because they were sampled from the same underlying populations. And if that is the case, then we can plug in the inverse variance weight into the formula I derived. But unfortunately, we are living in a non-idealized world. So the assumption I mentioned above can be violated in several ways. For example, the genetic ancestry between cohorts can be different, even if we label them as the same ancestry. Or at the same time, there could be like differences in experimental settings or measured phenotypes. Let's take depression as an example. So 23andMe use questionnaire-based diagnosis of depression, but some other cohort may use like clinical interview-based diagnosis of depression. So although we all name it as depression, but it is in fact correlated but different trait. So what will happen if we assume a fixed effect model, but when there are in fact between cohort heterogeneities? So we show that by simulations. So here the x-axis is the numbers of cohort included in the meta-analysis, and the y-axis is the SNP-based heritability in the meta-analysis. And the black dot here represent outcomes from simulated scenarios where SNP-based heritabilities varies across different cohorts and genetic correlation between cohorts are less than 1. And the color dot here is what we observed in the real data analysis. And from this plot here, we can show that our simulated scenarios perfectly explain what is observed in the real data analysis. And our simulation shows that when there is heterogeneities in cohort-specific SNP-based heritability, or when genetic correlations between cohorts are significantly less than 1, then SNP-based heritability estimate in meta-analysis can decrease as the numbers of being as the number included in the meta-analysis increases until reaching a plateau. And this is our conclusion number 1. And now let's focus on the out-of-sample prediction R-square, and let's go back to this slide. So the H-square in the discovery cohort is a theoretical upper limit of R-square. It's based on the assumption that both discovery cohort and the target cohort were sampled from the same underlying homogeneous populations. And similarly, SNP-based heritability in the meta-analysis is the theoretically upper limit of the R-square in the target cohort. Is based on the assumption that every single cohort involved in the meta-analysis and the target cohort were sampled from the same underlying homogeneous populations. But as I have shown previously, like this assumption can be violated in several ways. So here we derive conditions when R-square can be higher than H-square. So here the equation here means that the SNP-based heritability in the target cohort times the squared genetic correlation between the target cohort and the meta-analysis cohort. So when this quantity here is greater than the SNP-based heritability in the meta-analysis cohort plus an error term, then R-square can be higher than H-square. So ME here is the effective number of SNPs and in European populations, it is around like 60,000 based on previous studies. And this inequality here also explains that why in idealized situations, H-square should be the theoretical upper limit of R-square. It is because in idealized situations, SNP-based heritability in the target cohort should equals to SNP-based heritability in the meta-analysis cohort. And genetic correlation between the meta-analysis cohort and the target cohort should equal to 1. And then these two terms get canceled out. And because like 0 will never be greater than something greater than 0. So this inequality here will never hold in idealized situations. And with an increasing sample size, R-square will be approaching H-square, but R-square will be never higher than H-square. However, in non-idealized situations, R-square can be higher than H-square. And we show that by doing a leave one out meta-analysis using data from the PGC cohort. So what we are doing here is that with initial C+1 cohort, we leave one cohort out at a time, and then we meta-analyze the remaining C cohort. And with the resulting meta-analysis summary statistics, we are able to estimate the H-square, the genetic correlation between the meta-analysis cohort and the target cohort. And we are also able to generate genetic predictors with the meta-analysis summary statistics using methods like ISPSR and predict into the target cohort and get the out-of-sample prediction R-square. And this is the result we get. So here, x-axis is HT square times RG square, and y-axis is the out-of-sample prediction R square. So what we are claiming is that when HT square times RG square is greater than the predicted threshold here, then out-of-sample prediction R square will be higher than the SNP-based heritability in the meta-analysis used to generate genetic predictors. And this is exactly what we observed here. So let's take this cohort as an example. So when HT square times RG square is higher than the predicted threshold, then the out-of-sample prediction R square for this cohort is higher than the SNP-based heritability in the cohort that is used to generate the genetic predictors. And on the other hand, when HT square times RG square is lower than the predicted threshold, then the out-of-sample prediction R square in this cohort is lower than the SNP-based heritability in the meta-analysis. So this comes to our conclusion number 2, that when between cohort heterogeneity exists, R square can be higher than H square as long as the condition outlined here is satisfied. So now, bearing the heterogeneity ideas in mind, what we proposed is that in the future meta-analysis, what we could do is that we need to first define a focal trace. Let's take depression as an example. So we could take clinical interval-based diagnosis of depression as a focal trace. And then, like, instead of treating those proxy traits, for example, self-reported depression or GP record of depression as the same trait and fit them all in a fixed effect model, what we can do is to use tools like MTAG, which is a revised version of random effect meta-analysis, and kind of borrow the information from those proxy traits into the focal traits instead of fitting a fixed effect model. And we believe this could potentially increase the statistical power. And we have submitted a secondary analysis proposal to test this idea, and hopefully we will be able to get the result very soon. So we have published our work on AJHG this year. So if you want to look into further details, or if you have any questions, please feel free to refer to this paper or send us a message. And that's it for my presentation. Thank you so much. Any questions?
Thank you very much, Mark. I think it's clear that this work was very much motivated by our actual observations with the Psychiatric Genomics Consortium data. Mark has already made this presentation to the major depression group, but I thought the observations that we had are actually very relevant to many of the other disorders. So with that, are there any questions? Nice clapping coming up my screen. Any questions? I'm in this kind of panel room, so it's really hard to see what people are doing. I don't think we've got any hands up at the moment. Yeah. Okay. Well, in that case, we'll move on to the second talk, and then at the end of the second talk, if people have got questions for Mark, then they can throw those in later on. So the second presentation today is from Dr. Jian Zeng. So we call him JZ. He's based at the Institute for Molecular Biosciences at the University of Queensland. And he, if you know his work, he's got some very nice papers recently looking at using GWAS summary statistics in various ways and developing methods which describe genetic architecture from summary statistics and also risk prediction methods. And so JZ, I think, has got a new paper which hopefully will be coming out soon, and today he's going to share that work. Over to you, JZ.
Thank you, Naomi. It's my great pleasure to present this work. Good morning, afternoon, and evening, everyone. And today I'm going to present our latest research on polygenic risk prediction, incorporating functional annotations to the genome-wide coverage SNPs. And we're going to show the results within and between ancestry predictions. So just a minute introduction about polygenic risk scores. It predicts the genetic liability of an individual to disease. It can be simply calculated as the weighted count of the risk alleles. So PRS is a linear combination of the risk alleles of the SNPs across the genome, where in this equation, key questions includes which SNPs to be included in the prediction equation and what are their weights. The conventional approach picked genome-wide significant SNPs and used the weights SNP effect estimated from the GWAS study. And the more recent model tend to use the genome-wide SNPs and re-estimate the effect using the summary statistics and LD information. So although the PRS is very promising to identify individuals with high genetic risk, the current medical application has been limited by modest prediction accuracy in most complex diseases. As shown in this study, they looked at the prediction accuracy measured by AUC, the probability ranking a case higher than a control in a number of complex diseases. And the AUC is not very high in both of the cases. Another major problem is the GWAS study is biased towards the European ancestry. And when we do prediction across ancestries, the prediction accuracy decreased substantially. So it's a very important question how to improve the prediction accuracy in both cases. And we think having a knowledge about the causal variants and their effect will help to improve the prediction. But if the two SNPs are in high LD, it's very hard to distinguish which one is the causal variants using the genomic data alone. So the functional genomic annotation can help to provide such information. It has been shown previously that across different functional annotation categories, some of the categories are enriched in the SNP heritability. That suggests the causal effects distribution can be different between annotation categories. And this information can help us to identify the causal variants and capture their effects. So in this study, we developed a new methods to capture that. And the method is based on the previous developed methods called S-base-R. These slides briefly introduce what's S-base-R. So it's a method built on this generic model for summary statistic model method. Essentially, we are fitting the SNP joint effect as a linear combination of the GWAS SNP marginal effect. So on the left-hand side, this is the GWAS summary statistics, and we need the LD correlation matrix calculated from a reference sample. In S-base-R, we assume the SNP joint effect has a mixture prior distribution. So this is a mixture distribution of multiple components, including a point mass at 0 and a number of normal distribution with different variance. The reason we want to use this mixture prior is by using different mixing probabilities or the pi values in this mixture distribution, we can almost be able to account for any effective distribution and therefore accommodate various genetic architectures for the traits. And this slide just to reinforce why we use this mixture prior. Through years of research, our experience says that the best model for PRS is the model that best describes the distribution of the SNP effects. And indeed, for different traits, we found the genetic architecture can be quite different. Some of the traits are, most of the traits are dominated by the small effect that explains most of the genetic variants, but some of the traits, there are large effect contribute to the traits. So that's the benefits, and that's the reason we want to use the multi-component Gaussian mixture to account for these various genetic architecture. So in our new methods, we incorporate the functional annotations. In S-base-R, this mixture distribution is identical between SNPs. And now we are going to allow this mixture distribution to be depends on the annotation that the SNPs received. And the way we do that is to incorporate the SNP annotation into these mixing probability parameters pi's and allow the pi to be a function of the SNP annotation. So this is like a generalized linear model for the membership of the SNP to different distribution components. The interpretation of this model is that if the SNP annotation is biologically important to the trait, then that should increase the probability of the SNP being a causal variance, or increase the probability of the SNP has a larger causal effect. And we estimate both annotation effect and the SNP effect in the model from the GWAS data. Another major improvement from this method is that we employed a low rank model such that we are able to analyze many imputed causal variants simultaneously. In this study, we fit up to 7 million SNPs in the model. The way we do that is to first partition the whole genome into about 600 approximately independent LD blocks. And in each block, we calculate the LD correlation matrix. Then we do eigen decomposition on this LD correlation matrix to have the eigen vector matrix, a diagonal matrix of eigen values and the transpose of the eigen vector matrix. Then the trick is, instead of using all the eigenvectors, eigenvalues, we only use the eigenvectors correspond to the top eigenvalues in this LD matrix. And then follow up with the simple transformation of the model, we can end up with the new equation, where in this equation, we are not fitting all the marginal snip effect in the model. Instead, we are only fitting a proportion of them. And that substantially decreased the dimension of the equation. But note that we still fit in all of the snip joint effect, so that we can incorporate the annotation model as I described previously. So it turns out we don't have to keep many eigenvalues in the model. We can only use the top 20% of the PCs, the principal components, that are enough to explain 99.5% of the variance in LD in the human genome. So that means we can get rid of lots of rows in the equation without a negligible loss of accuracy. So of course, we do simulation to test the robustness of our methods, but due to the time, I'm just going to show the real data analysis. So we analyzed a number of GWAS data sets, including UK Biobank, FinGene, LifeLens, Page, and Jian data sets. And in the UK Biobank, we have data across different ancestries, including European, East Asian, South Asian, and African ancestry. We use the two SNP panels. One is 1 million HiMAP3 SNPs. Another panel is 7 million imputed common SNPs. They are all with minor allele frequency greater than 1%. The annotation data from Gazelle et al. 2017 paper, the LD score regression baseline LD model, they have curated 96 genomic annotations, including binary and quantitative annotations. And we compared to a number of methods. We found the best methods in the market. Some of them also incorporate annotations in the model, such as MegaPRS and LD-PREP-FUNCT. So here's the results for the within European ancestry prediction accuracy. As shown in the y-axis, we looked at the prediction accuracy from different methods, and they all compared to the S-base R using 1 million HiMAP3 SNPs without annotation as the benchmark. So the y-axis shows the percentage of the improvement of the prediction accuracy compared to S-base R. And the x-axis shows the different combination of the SNP density and the use of annotation. So we have 1 million SNPs without annotation and 7 million SNPs without or with annotation. As you can see, our method S-base RC in purple can increase the prediction accuracy up to 14% compared to S-base R. And it also outperforms the other methods, including LD-PREP2, LD-PREP-FUNCT, MegaPRS, and the later two methods also incorporate functional annotations, but in slightly different ways. And this is also within European population prediction, but predicting to a cross biobank. So on the left panel, we are using the FinGene data as training and predicting to the UK Biobank. And on the right-hand side, we use the published available GWAS from meta-analysis and predict into the same traits in Lifelines Biobank. And in both cases, UK Biobank, European data was used as the LD reference. And now you can see the S-base RC demonstrated an even larger advantage than the other methods. This is likely because the use of the low rank model in S-base RC, where when we remove the the small eigenvalues, we're likely to remove more variations, sampling variations in LD, because the small eigenvalues are more likely to capture those chance variations, and that can improve the model robustness. This is the result for chance ancestry prediction. So here we are training the model in European and predicting to South Asian, East Asian, and African population. And we found that the S-base-RC can improve the prediction accuracy by up to 34% in African, but also significantly improved in other ancestries. And here we compare to polyploid S. This is the method published last year. They also incorporate functional notation in the file mapping as the first step for prediction. But our method is a unified, coherent method estimating the annotation and the SNP effect in one model. So we provide a one-stop solution to estimate the SNP effect incorporating annotations, and it turns out to be better than the stepwise approach as shown in polyploid S. And here we compare to another method called the PRS-CSX. This method does not use annotations. Instead, they incorporate information from another GWAS cohort in the same ancestry with the target population. So in this study, we used the UK Biobank European GWAS data and the Japan Biobank sample to predict into the UK Biobank East Asian samples. And using these methods, we found that it improved the prediction accuracy by 17% compared to our benchmark S-base-R. So how does that compare to using annotations? Well, using S-base-RC without the extra GWAS cohorts, but with annotations and 7 million SNPs, we found it improved the prediction accuracy by 17%. It's comparable to what we found with PRS-CS method. Now, if we combine both sources of the information, the functional annotation and the extra GWAS cohort from the same ancestry as the target cohort, we observed an improvement of 33%, and this is larger than using either extra GWAS data or the functional annotations. That means if we want to maximize the prediction accuracy, we should use all the data available. And this shows the same thing, but here we use the page data, which is the mixed African ancestry, and predicting the UK Biobank African population. In this case, PRS-CSX improved accuracy, but not much. But here, when we combine both functional annotation and extra GWAS data, we observed the improvement of 39%. Another interesting result we observed in this study is the significant interaction between SNP density and annotation information. You might have noticed that using the 7 million SNPs and annotations results in the prediction accuracy higher than using the information from either source of the information alone. And here, this graph explicitly demonstrates that. So the x-axis shows the relative prediction accuracy using annotation versus not using annotation at 1 million HapMap3 SNPs. And the y-axis shows the same thing, but at the 7 million imputed SNPs. So if the improvement of the prediction accuracy using annotation is independent of the SNP density, we should be able to observe the results sitting on the diagonal line. However, here, all of the results are above a diagonal line, suggesting that the improvement of the prediction accuracy using annotation is higher when using a high-density SNPs than as compared to 1 million HapMap3 SNPs. And it turns out the reason is not hard to understand. It is because the SNP markers can tag the causal variants by LD, but they may not tag the causal variants by annotation. They may not share the same annotation as the causal variants. So if you use the low density SNP panel, the causal variants some of them are likely to be unobserved. And in that case, there may be a mismatch between the causal effect and the annotation. So these results also suggest that it might be beneficial when we use the whole genome sequence variants for prediction with annotations on them. We have a preprint, and this paper has been accepted in principle to publish in Nature Genetics. In terms of methodology, we have two major advancements. One is we developed a low-rank model that can fit a high number of SNPs with the genome coverage to better model the LD, and that could also improve the robustness of the methods in terms of the LD differences between cohorts. Secondly, we incorporated function annotations to better capture the causal variants and their effects. So in terms of science, in summary, these methods improve the prediction accuracy by 14% in European ancestry and by up to 39% in chance ancestry prediction. It do a better job than the other methods, the best methods we found in the market. For chance ancestry prediction, the function annotations with genome coverage can provide comparable information as using extra GWAS datasets. And to maximize the prediction accuracy, we should use both sources of information because they tend to provide additive information. And we also identified a significant interaction between SNP density and the annotation information that suggests using the whole genome sequence variants in the future might further improve prediction accuracy. And there are more results from this work, but I don't have time to show all of them. Please feel free to check our paper. And at the end, I'd like to thank Zili. He's the first author of the paper, and my mentors, Peter Vischer and Naomi Wray, and all of the members from our group. And I also like to thank the data consortium and the funding bodies. Thanks everyone for your attention, and I'm happy to take questions.
Thank you, Jay-Z. That was another very beautiful presentation. Grace, how do people actually ask questions using the microphone rather than just in the question and answer? Well, Grace is telling me that. Grace is saying, please raise your hand. But Andrew McIntosh, Andrew, do you want to say it out loud? Are you allowed to do that? If not, his question is, you may have said, but how does SBayes-RC handle the MHC region? What happens to SNPs in that region? Good question.
Yeah, thanks for the question, Andrew. For MHC, we include that into the analysis. We include the blocks that contains the MHC, and we apply the same strategy as the other regions. We calculate the LD matrix and do the eigen decomposition in that block. Because of the extensive LD in that region, I think we might remove quite a bit PCs from that region, but we use the top 20% PC to capture the major LD patterns. And I guess that could help to analyze the data in that region, because we know the LD differences might tend to be larger due to sampling variation between like the GWAS cohort and the LD reference cohort in that region. And when we do this eigen decomposition and only select the top 20 PC, that could help to remove some of the chance LD. This is also something new in our method. In the previous SBayes-R method, we removed that regions to improve the robustness, but in the new analysis, we can keep them.
So while other people are thinking of questions, so Jay-Z, with S-Base-R, I know that sometimes people have used it and found convergence problems, or particular GWAS data sets have kind of funny properties. Can you just say what you've done in S-Base-RC to try and help the robustness?
Yeah, thanks, Naomi. Yes, this is a common problem found by ourselves and other peoples. When you run S-Base-R, especially you analyze the data from meta-analysis in the public domain, it's likely to be sometimes they deal with convergence problem. And we think the problem mainly comes from 2 points. One is there is LD differences between the GWAS and the LD reference samples. And secondly, when you do the meta-analysis, there might be some cohorts that SNP has no genotypes. So the per SNP sample size is not the same across all SNPs in the meta-analysis. And for the first one, the LD differences, the new methods use this low rank approximation. So that only pick the top pieces and remove the pieces with small eigenvalues. And that could help to remove the chance LD due to the samplings sampling errors between the two cohorts. And for the second one, we suggest you to look at the per SNP distribution first, and then to remove the SNPs with the per SNP sample size as the outlier, like the 3 or 4 standard deviations to the median value. And in the new method, S-Base-RC, we have included this QC step in our pipeline. But sometimes you see a funny distribution like a very long tail, or even a multimodal distribution for the SNP effect, for the SNP sample size. You had better to check that prior to the analysis as well.
Thanks, Jay-Z. And I think it's fair to say usually those problems occur with kind of meta-analyzed data sets rather than the kind of UK Biobank type single cohort data sets. Is that right?
Yes, yes. And it seems to be a problem to all of the summary database, the Bayesian methods. As I showed in one of the results, we looked at prediction accuracy across biobank, and the other methods suffer this problem so that they have to decrease the prediction accuracy. So my experience is no matter what method you use, you need to check those things as a QC step. But our method is tend to be more robust to these errors.
Okay, thanks, Jay-Z. So there's a question for you in the chat. I'll maybe move to the questions for Mark while you read that question. So Mark, can you come on? Jay-Z, I just thought you might want to take a bit of time to read the question from Paula. So Mark, there are a couple of questions for you. One of the questions was, how does the approach apply to heterogeneity between the sexes?
Yeah, so thanks, Marcos, for proposing this question, and prepare a quick slide. So yes, it does apply to heterogeneity between sexes, and here I've presented a quick example. So for example, if there's a trait where SNP-based heritability in both male and female equals to H squared, but there are some like sex-specific effects such that the genetic correlations between male and female are actually 0.5, then like based on the formula here, so if you put both male and female cohort together, you will get a SNP-based heritability estimate at like 0.75 H squared, although we know that the SNP-based heritability in both male and females all equals to H squared. But also it's noticeable that for most traits, RG between male and female equals to 1. But for those traits that has sex-specific effects, so you need to consider the heterogeneity here. And it is also kind of relevant to the second question. So the second question asked me about like how to get the like a reliable SNP-based heritability estimate. So the estimation error of SNP-based heritability depends on the method used. For example, for method like GRAML, the estimation error can be approximated by this formula here. So 679 over the total sample size. So how did I get this magic number here? You can just refer to either GRAML paper or our paper. But what this shows here is that with an increasing sample size, you get a smaller and smaller estimation error. But as I mentioned here, that it is one thing to consider the estimation error, but you need also to consider like between cohort genetic correlations. For example, for depression, I think depression defined based on clinical diagnosis based depression is quite different from depression based on questionnaire diagnosis of depression. So yeah, hopefully this answers your questions.
Thank you, Mark.
Yeah.
Okay, Jay-Z, would you like to answer the question that Paula's?
Yeah, I'm trying to answer Paula's question. Thank you for the interesting question, Paula. Yeah, for the first one, how do we combine the two populations data in the S-based RC? Yes, we estimate the eigen decomposition data for the other population as well. So basically, what we were doing is we run S-based RC separately in the two population using their own LD information. And then we have a tuning sample with individual level genotypes and phenotype to combine the SNP estimates and generate the final weights for the target population. So this is different from what PRSCSX is doing. They analyze both GWAS data sets in one model and modeling the genetic correlation between them. But we are taking a simpler approach and to do that, but it already shows that the benefits of combining two data sets together with the annotations. For the second question, if the variance in the intronic region with significant effect, how do we deal with that? In S-based RC, we analyze one trait at a time, and the annotation effects on the trait genetic architecture is also estimated jointly with the SNP effect. So that means the annotation effect is estimated in the model and also specific to the trait. For that trait, if the intronic region is important, then it will have a higher weight. However, if only a few SNPs are important in intronic region, those SNPs might likely to receive the other annotation as well. And our method allows for the overlap between annotations. And those SNPs will be upweighted if the sum of the importance of the annotation are put together and have the SNP effect upweighted.
Thanks, Jay-Z. While we're waiting for someone else to ask a question, in your results, you're doing analysis of many genome-wide association studies, and you're presenting, the average and the distribution across those. I was wondering if you have a feeling whether S-based RC is particularly good for traits which are very the impact of polygenicity. So, for example, your own results have shown that disorders of the traits of the brain are much more polygenic than many other traits. So would you expect to be such a good increase for S-based RC for those very polygenic brain disorder traits compared to other traits?
Yes, thanks, Naomi, for the question. I think these methods works best if there is a complex genetic architecture, such as the presence of the small effect and also the presence of medium effect and large effects. And we know that for the brain traits, it has a large polygenic background gene effect, but also there are some large effects as well, especially when you move towards the low frequency. So I expect it will be particularly useful when we, in the future, include even the low frequency or rare variants in the model. And this model would be flexible to capture both the large effect at the rare variants and also the small effect at the common variants.
Good, thank you. Okay, any more questions? Don't be shy. It's very funny for us being locked in this host box. We can't see anybody else's reactions. Okay, well, in that case, we're nearly up to the hour anyway, so I'll just end by saying thank you very much to both Xiaotong Wang and Jian Zeng. I think you'll all agree with me that they both did really clear presentations on topics which I think are of interest to our Psychiatric Genomics Consortium community. So lots of claps going up here, so thanks very much. And we'll see you at the next PGC Worldwide Lab webinar. Thank you.
Thank you, everyone. Thanks, Naomi.
Yeah, thank you.